Hello, everyone. It's the top of the hour, so we're going to get started. Welcome to Lesson 12 of Certara.R School. I am glad that you could join us today. I am Anna Henry, Head of Certara Education. The session today will focus on the history and advantages of the pharmacometrics language PML. If you have any questions, please type them in the Q&A panel. It is my pleasure to introduce today's speakers. Dr. Michael Dunleavy is joining us from retirement to provide us the history and background on the development of PML, the pharmacometrics modeling language, which is the underlying language of R-Speaks and LME, RS and LME, Piranha and LME, Phoenix with Nutland, and Phoenix and LME. And as you can see from his impressive bio, Dr. Dunleavy has a wealth of experience in computer science and pharmacometrics. Among his many accomplishments is the fact that he was the creator of this exceptional language, PML. We are excited that Dr. Dunleavy has agreed to join us today so we can all gain a better understanding of how the PML language was developed, what problems it solves, and advantages over other languages. Joining Dr. Dunleavy is also Dr. Keith Nearforth, current product owner for Certara's NLME, Piranha, and Trial Simulator. Dr. Nearforth will also provide us several examples of the power of the PML language during his presentation. So without delay, I give the floor to Dr. Nearforth to get us started.
Great, thanks, Anna. So, hi everyone, welcome to another edition of Certara.R School. We're very happy you've joined us today. I wanted to start today's lecture with a history of PML. I think it became an object in Mike's mind sometime around the 2002 timeframe when Trial Simulator was originally launched. So Trial Simulator, as you may know, is a clinical trial simulation tool. It's written in C++. There is no underlying pharmacometrics language to it. Rather, it uses a graphical modeler and then runs simulations by compiling or preparing Fortran files. And this is when Mike started to think about a language that could be transferable. In other words, used for an analysis and then pressed directly into a clinical trial simulation. In 2005 was when the development of the Phoenix platform started, with 2009 seeing the release of Phoenix with Phoenix NLME. The underlying language of Phoenix NLME at that point was PML, and that enjoyed a relatively, stable, large user base for many years. Fast forward to 2020, when I joined software, we were seeing an explosion of the use of R within the PMX domain, people supplementing a variety of analysis engines with R-based solutions. And at that point, thought to myself, well, this is being done. The nature of the domain is use of multiple tool sets. I think it makes sense to build professional solutions that are R-based, so that we can maintain, support, and also train users on the use of these tools, hence, contributing to their career development. This is the whole concept behind RSLME and Certara.R School. In 2021, we integrated RSLME with Piranha. Again, Piranha being a tool that's able to harmonize use of underlying flexible tool sets without sacrificing that flexibility. And then finally, later this year, we're going to be introducing an R package that allows you to use PyDarwin, automated machine learning, with PML and NLME. Now, at several points during my work in this field, I've been quite surprised that when we think of something new to do, we look at PML, and the answer is, yeah, PML can do that. And I think to myself on several occasions, how did they know back in 2002 to design this language this way? Because we have not touched the underlying PML language, yet we continue to use it for increasingly innovative solutions in this domain. So we were at a team meeting, and I said, it would be interesting to speak to the person who actually built this language, to understand what they were thinking, and have them come to Certara.R School and tell a story. that's how this lecture developed. At this point, I'm pleased to turn this over to the creator of PML, Mike Dunleavy. I think today's talk might be a little bit less technical, but I hope you enjoy the lesson. Thanks, Anna, and over to you, Mike.
Hi, everybody. This is Mike Dunleavy. I've been around computers since the late 60s, early 60s. And so if you're familiar at all with computer science, the idea of creating a language is not a big deal. I was introduced to the pharmacometric world back in the 90s, and was with the graphical user interface. And the point of a graphical user interface is that there's data underneath, and the data is naturally expresses the problem. But then if you put the two together, you get a declarative language, or what I call a problem-oriented language. So if I can just continue here. The idea is that the source code should express the problem for which you're trying to solve. And there can be multiple solutions to the problem. Alternative languages require the source code to be changed whenever the solution changes. But if you want to, for example, if you want to change from an ordinary differential equation solver like Runge-Kutta to matrix exponent, you don't have to alter the problem-oriented language at all, or be bothered with doing it correctly. If I can emphasize that point, there's a lot of math involved here, but it's nice if you can just call it up and expect it to be correct. Another thing you want to do with a declarative language or problem-oriented language is you may want to add things to it, like what is the number of headaches a subject had this week. In PML, you would just say count, as opposed to, gee, the log likelihood of getting 6 headaches this week is such and so on, big calculation. You can get that with just a simple one-liner. And if you need to say, include a zero inflation like they sometimes do, that's also something you can easily add by just saying that the zero probability is the inverse logit, where the logit is a mathematical function, where E is some expression. And the nice thing about logit is that it's between 0 and 1, so it's a great probability. And then if you need to do any trial simulation, if you fitted a model using NLME, then you can just take it and use it directly in a trial simulation without rewriting it. So if you can go to the next slide, Anna. Okay, so if I can just digress a little bit about what makes a declarative language valuable. There are thoughts in your head, and they are real. They are in a data structure, and every data structure contains nouns, verbs, and adjectives that represent real world things. And if you just listen to two pharmacometricians talking to each other, you can easily get a, if you record that, you can easily see the nouns, verbs, and adjectives being used. And that's what the declarative language should contain. It requires that declarative computer language is one that lets such things be communicated to a computer as well as to another person. So that a measure of that is when you change your mind in a small way, the corresponding change to the code is small. What that means is it's not easy to make a mistake, because when a program written in a computer language fails, it's because more than one change was made to it and the changes don't agree with each other. But if you only have to make one change, it's pretty hard to make a mistake. It may not agree with what's in your head, but that's a separate issue. That doesn't mean, if it's a declarative language, that doesn't mean it has no programming constructs like procedures and if statements. It depends on what you're trying to convey. And I will see an example of that later. So if you can move forward there. So here's an example of what it means to describe a pharmacometric model. Suppose you have a very simple model of systemic blood represented, and the amount of drug in it is represented by the letter A, and the volume of it is represented by the letter V. There's an elimination rate, a KE, which is in 1 over time, and that amount, that tells you the rate at which the drug decreases from the systemic blood. If the systemic blood is a point where medication can be introduced by means of, say, an injection, then we would say that a dose goes into A. It might not, but in this case, in the simple case, it does. So if we go up and look at the statements on the left, we can write a differential equation that says the change in the change in ADT is the amount A times the elimination rate. And if we say that A is where we want to have doses, then we say dose point A. And that gives us a point where we can hang additional information like time lag and stuff like that. Now, KE is what we call a structural parameter, meaning it's a parameter of the structural model, which is on the right. And we want to say, well, it depends on a fixed effect and a random effect. And here the fixed effect means that the typical value of KE is such and so for all the people in a population. And the letter N is the closest thing in English to the Greek letter eta. So I say that the eta is the amount, the eta KE is the amount by which it varies per individual, because every individual will have an independent KE. So the way we say that the typical value of KE is a fixed effect is by the next line, and it says the initial value is such and so, and we have room in there to provide limits. If you don't want to give the limits, then you can just leave them blank. In addition, we want to say something. We want to say that eta KE is a random effect. That's the next line, and the 0.1 is the initial value of the variance of KE. And if anybody wants at the end to ask what's variance, I'll be glad to answer that. Now, you can't observe the amount of drug in the systemic blood, but you can observe the concentration. So in order to see the concentration, we have to divide the amount by volume, the volume of systemic blood, and that becomes another structural parameter, which has a typical value and an eta. And then finally, the last line, in order to observe the concentration, we have to say, well, we got an observation, but it's not correct. It's off a little bit by an epsilon, and the epsilon itself is another parameter for which there can be a variance. In this case, the initial value of the variance is 0.1. Now, there are plenty more complicated ways we can write these statements, but this is the very simplest. Okay, you can move ahead. This shows what happens during a during a model fitting. The first thing, okay, NLME 7 is the nonlinear mixed effect program. And the reason it's 7 is that there was a 6, and before that there was a 5, and that's just the version. There may be an 8 now, I don't know. But in any case, it receives the actual data that you're trying to produce the model of, and a set of column definitions. Those are two files. And the first thing we do is take the data and encode it as bytecodes, so we're not trying to make decisions all the time as the thing runs, as to, for instance, if an observation and a dose happen at the same time, well, which happens first? So by putting it as bytecodes, we make that part efficient. At the very end, once we've found all the observations and ADAs, we can generate output. So the optimization itself, Bob Leary had wrote a bunch of fitting algorithms of various properties, and but they all kind of work by the first thing you got to do is find the best. The outer loop is you got to find the best fixed effects, the thetas. And at a particular choice of thetas, you got to find the best ADAs for each subject. And in order to find the best ADAs for a subject, you have to get the log likelihood for that subject, and by running the model. And so what happens is that the PML model is translated into C, and it becomes a dynamically linked library. And NLME7 loads that dynamically linked library, so it's as fast as possible. So what we do for that is say for each subject, and we can parallelize over subjects, which again, we can thank Bob for, to get maximum performance. And for each subject, there's a series of time segments in the data. And between time segments, we run the ordinary differential equation solver. Now, there are some models which are not time-based, and you know they're not time-based because they don't have any differential equations, in which case those for each time segment and run ODE solver out don't do anything. If that's okay, let's see, let's move on. Okay, so here's an example of the kind of thing you can do with declarative information. You can say, what will we see at steady state? You know what steady state is. You assume we've been giving the doses for an infinite length of time in the past, and so the system is at steady state. And so how do we do that? Well, we don't have to change the model at all. What we can do is add something to the column definition file that says there's a steady state, there's a row, there's a column where if there's a 1 there that says that's a steady state. And if you see that, that means assume there's a bolus to the A1 compartment of 10 units and of medication, followed by a wait for 24 time units. Notice I say time units. It could be minutes, could be hours, could be months. Doesn't matter because the math is the same. So suppose you do that, then it automatically puts the system into steady state, and then you can do observations. Now notice there are some kind of compartments for which there is no steady state. And so there's special handling for urine compartments. I mean, the idea of a urine compartment is that it builds up forever until you observe it, and then it goes back to 0. So, so the steady state only applies to compartments that can actually work. And so if after you put that steady state in the data file, then you can make observations and see, well, what does it do? Just an example of a declarative language. Okay, forward. Here's another example of, it's easy enough to say, okay, we're going to observe concentration or something, and there's going to be an epsilon. But what if we're trying to observe something like the number of headaches you had last week? So that would be just in PML, you just say that's not an observation, that's a count. And a count means you observe a variable and there's a hazard expression. Let me say a hazard, suppose you on average have one headache per day. Okay, that's 24 time units. So the hazard expression be 1/24. And then so then you could say, well, the number of the number of headaches over the last week is the accumulation of the hazard expression over that week, and that's the average number for something like a Poisson distribution. In any case, there's lots of options. And what you see down below is what you would have to write if you didn't have the count statement. And again, you'd have to get the math correct. So let's ignore that unless somebody's interested. Okay, next. Normally, statements in a PML model apply all the time. In other words, if you have a differential equation, it applies to all points in time. And if you have statements that saying that you have a structural parameter, especially if it's a function of subject covariates like weight and age, and whether or not somebody is a smoker, those statements are true all the time, unless assuming it's a time-based model. But if you want to say, if you have something that happens at discrete points in time, independent of the actual data, then you can have a sequence statement, which means that within the sequence statement, the statements actually happen one after another, and time may be taken up. So for instance, enterohepatic recirculation is modeled here. You have a rate at which, say, a medication goes from the small intestine into the gallbladder, and then all of a sudden, say once every hour, and it may take 10 minutes, the gallbladder empties back into the small intestine. So how do you express that? Well, you can do it. This says do this 10 times and allow the fluid to build up in the bile fills up in the gallbladder. And so we say that the rate of flow from gallbladder to small intestine is 0 for a certain length of time. And then we allow the reflux to happen. We say Q is the amount of bile divided by the time reflux. And so the flow rate goes back into the small intestine over a period of time that may take a minute. So let's see if I can go forward here. Okay, so once you've gotten a model, how do you use it for something like modeling a trial? The idea in trial simulation is you want to know the power of a trial, so you need to do it many times. And so, for instance, if the power of the trial is 80%, so you can run the trial 100 times with random variations in the population, and if it succeeds 80 times out of 100, that's pretty good. You get a power of 80. So what does that consist of? There's a population model which generates subjects, and here's an example of it on the right. And so, for instance, this particular population model generates whether or not there are males or females and what are their weights and ages. And I won't go into what goes where the ... goes. then you can take the PK, and if you want, there's PD, the PD model might be separate. You can have any number of models here. But typically there's one or two. And then, well, how do you connect them all? Then there's a join statement that joins them all together that says that the covariate weight comes from the population and goes into the PK model, and the concentration goes into the PD model. And so those are connected. Then we have time-based operations. In other words, there's the treatment schedule and the observation schedule. And then what does a replicate consist of? Remember, some subjects get placebo or no dose, and others get dose. Then once you, so that takes care of one study, but if you do that many times, you can get the number of replicates of the study. And then you can say, well, what if we were wrong about how effective the medication is? If you suppose the medication was more effective, well, if you know that, then you might be able to get the same power with a smaller number of subjects. So if we look at the next page, these are examples of the kinds of procedures that you can use in a trial simulation, where you can say to do a subject, we just start the subject, means make up the gender and the weight and the age, and possibly discard the subject because they're outside, because they're too heavy or whatever. Then there are the procedures for treatments and procedures for observations. When we start those, we start them in parallel. The treatments happen in parallel, the observations happen in parallel, and then we just wait until they're done. So this is something that the procedures have. They can start as another procedure, and they can not wait, or they can wait. If they want to wait, they say call the treatments. And okay, so the treatments are a procedure. In this case, it's very simple. You give a single dose to the AA compartment of the PK1 model. And then the process, and then the observations are, we can sleep for 0 time. That means, and the 1 means don't wait. You take an observation right away, but take it after the bolus. That's what the 1 stands for. And what do we do at that point? We observe the concentration and we observe pain relief in the PD model. And then we wait for 1/4 of a time unit and do it again, and then wait for 1 1/4 time units and do it again. That's can be any process involved here. Excuse me. Then how about a replicate? Procedure for a replicate is, well, we have the placebo arm, which has a dose of 0 times the dose factor, and the normal process, which is we give a dose of 10 times the dose factor, where the dose factor is whatever it is. Excuse me. Then, okay, that takes care of 1 replicate. So if we want to do a bunch of replicates, like 100, fine, we can do them. But that would be 1 scenario. We call that scenario 0, where we have a normal process of running replicates. But in scenario 1, we might want to say that the medication is more likely to produce a result, in which case we have, say, an EC50 of 0.5, and then again, we do 100 replicates. And so that shows how we do scenarios. And finally, there is a top level procedure called main, which does everything, which does the scenario 0. And then you see the call. means wait for it to be done. And then the call, scenario 1, wait for it to be done. And that shows how this is just one simple example of how you can use this for trial simulation. If you need to, say, run the arms of the subject in parallel, then you can use start instead of call for the subjects. And there's a lot of ways it can be made more complicated, but let me stop here and let Keith continue.
Great, Mike. Thanks very much. So I hope that gives folks a sense for kind of the thought process behind the design of the PML language. We use it for modeling, but it can do so much more. And we intend to take advantage of that as we move forward with future development initiatives. But yeah, what I wanted to do is basically provide an example that demonstrates some of the points that Mike was talking about. Before I do that, though, I just kind of wanted to tie things together a bit in terms of Mike mentioned declarative language, which PML is. If you look at computer languages, right, there's a scope of computer languages that you can think about as this triangle here. Where at the very bottom, you have these low-level languages. These are languages where you're actually speaking to individuals.
Excuse me, I'm sorry to interrupt, Keith. I can't view your screen. I just want to confirm that other attendees can view your screen.
Sorry about that. How are we doing now?
Perfect.
Yeah, apologize for that. Excuse me. So again, you can think of computer languages on the scale from low level to high level. And with anything, there's pros and cons. Low level languages, really there you're speaking to, they're machine specific. You're managing memory at the same time. You're trying to instruct these systems to do what they're doing. And at the very lowest level, they're not even human readable. But they're fast. They're very fast. Whereas at the top end, you have the declarative languages like PML, Python, R. These are human readable. And essentially, as Mike mentioned, these declarative languages focus on describing what to do, not so much how to do. But there's give and take. To look at like an example that really puts this in context, I think, is to ask, how would you write a function for calculating a mean? So in my mind, right, I'd have X as I imagine to be some vector of numbers, right? And to calculate mean, I'd take the sum of those numbers and divide by the length and get a mean. When you look at a declarative language, what it does is it tries to anticipate use cases and adapts to variations in the problem space. And in the end is more generally useful across, a large number of use cases. And if we look at the actual code for mean in R, which you can do by this get anywhere mean default function here, you see that the mean does more than what I imagined, right? First, it checks the input. Are you giving me a vector of numbers, right? If they're complex, it returns an error. Wait a minute, I can't calculate mean on this. So it anticipates that perhaps the wrong input might be coming in. It says, okay, I'm going to check for NAs, right? And if there's NAs, how do I handle them? There's an option for trim mean. I personally have never used a trim mean, but I guess it's common enough in statistics that they've built that into the R function for mean. And then finally, the R function calls some internal C function that actually does the calculations for the means. So again, you can see how this declarative R language has anticipated a bunch more use cases than perhaps what I would have done. Just a good example for really what a declarative language is. The final thing I wanted to do is kind of touch on this balance between capability and complexity or ease of use that we consider here as we pursue further software development. And if you think of ease of use, right, it's the reciprocal of complexity. And think of capability as the ability to handle unique analysis needs. Now, this is something that's very common in the PMX domain, and I believe, it's the reason why, if you look 40 years back, this domain has always been characterized by the use of multiple tool sets. Because we are pharmacometricians, right? You can imagine a effective capability 90 value, that being, okay, you want your tools to have this ECAP 90 value. In other words, the functionality or the capability required to do the full job, right? So the goal in software development, as we try to make our tools better and better, is to shift this curve to the left. We want more capability for a given level of complexity or ease of use. Now, how do we do this, right? Well, one approach is to build a GUI, right? And a GUI, in fact, gives you a lot in terms of ease of use. But when you build GUIs, you take away some of the flexibility, and you also may, for a given analysis problem, not be able to reach your ECAP 90 for that particular problem. Next thing you might do is a good declarative language like PML. And sure enough, you can shift that curve to the left, just like you do with the GUI. However, you sacrifice some in terms of ease of use, but you still might get to your ECAP 90, but you're still sacrificing a bit on the top in terms of reaching the maximum capability compared to a completely flexible, command line language. So, what's a software company to do? Well, what we've done is we have Phoenix, NLME. It's a GUI environment. It's easy to use. It has pretty strong capabilities, right? But like any GUI, there are problems where you reach the limits of that GUI. And what we've done is we've built RSNLME to provide that bridge into the range of capabilities that can't be reached with a GUI tool. And we've done that in a professional supported way so that we can train and maintain these solutions moving forward. You can imagine there's a hard line here. You can imagine the shaded area actually coming down into the Phoenix, right? Because you can do anything with RSNLME. And you can also imagine over time, our goal is to push the capabilities of Phoenix up higher, right? But it's a good representation for why we've done what we've done. Finally, on top of that, we've built R-based tools that are agnostic to the analysis engine to capture that, that top level of capability. And in the end, right, we maximize the capability by supporting these different, ranges of capability, all with the same PML language. Again, this works throughout the whole scope, and we support both. So I hope this gives you a really good feeling for why we've done what we've done with RSNLME and Phoenix. This is what we're trying to achieve. Push this curve to the left while maximizing the capabilities of our collective tool sets. So for my example, I have, again, I tried to think of a representative example that would demonstrate what Mike is talking about in terms of having a thought and then being able to implement it quite easily in PML. So we're going to do a steady state simulation from a single dose model fit. The thought being, all right, I have my model. What concentrations will I see with a Q12-hour dose schedule? And the way we implement this is by adding a single column to the data set, and we'll call that column SS. And then we use this function, which we haven't seen before in our school. It's called add dose cycle. And this basically is used to specify a dose schedule of interest for this steady state record, right? Now, add dose cycle, I'm going to use this for a steady state example, but it can also be used for additional dose records. There you'd add an ADDL column. I have an example of that in the code. If I have time, we can cover it. So add the dose cycle, run the simulation, plot the results. We've covered running the simulation and plotting results before, so I'll kind of breeze through that in the interest of time. But again, what this function is doing, if we take a closer look, is add dose cycle. We specify a type steady state, and that's saying, okay, I'm going to add a dose instruction to my model that uses steady state. The input amount from this column amount for this simulation is going to come from my amount column. Now, the cool thing is that you could specify a volume here, a value here. So my amount in my data set is 5,000. I could put 10,000 here as a number, and then I'd get a steady state profile for a dose of 10,000. So you can either map or use numbers in this command, which demonstrates, again, the flexibility of the language. The amount will be placed in compartment AA. My column name that's going to trigger this is steady state in the data set, and my interdose interval is 12. And again, interdose interval, if you wanted to, you could put this in your data set and say II equals, II in my data set. So it's flexible, whether you map to a column or specify a number. I hope that's clear. It can be maybe a little confusing, but we'll see how that's actually implemented under the hood. Mike mentioned the column mappings, or otherwise known as the column definitions file, right? To date in our school, we've really kind of looked at this in terms of mapping model variables to the data set. Like what are these names in the data set and how am I using in these models? And when you submit a model to NLME, as Mike touched on, right, what are you actually submitting? Well, you have your PML, a data set, and your column mappings. Now, the cool thing about column mappings is it could be used for more than just mapping your data set variables to your model. Here we see an example of that in a single dose fit, right? You see our mappings, but you also see this table statement. Well, what is that? Well, that's the default table that you get from fitting a single dose model. By default, it's in there. Now, when we use our add dose cycle command, and we'll look at this in R, what is that actually doing? Well, under the hood, it's going to the column definitions file and inserting this row. And that should look familiar from Mike's slide. That's where that syntax is going. It's saying, okay, there's a steady state dose record, the column name is steady state, the amount is amount, and it's going to be a bolus into the AA compartment with a Q12 repeat cycle. And it looks for, again, this steady state. That's all you have to add to the data set, and that add dose cycle command. So right now I'm going to swap over to R, and we'll go through the example. I think I have 20 minutes left. That's great. So a lot of this will be overview, right? So here I have my R project code. I'm going to load up my libraries. I'm going to read my data set in from lesson 2. And of course, this example is completely reproducible. James showed how you could download all of the R school lesson and code from GitHub to rerun it. So if you want to repeat this yourselves, that's quite easy to do. So we're going to bring that data set in. I'm going to define a PK model. This is just a first order KA one compartment model with a weight effect on clearance. We can print that model, and then we fit that model. Now I've already fit this model, so in the interest of time for the lecture, after we fit that model, we can generate the exposed database. This should all be review, and then we can get our our diagnostic plots out of this. So it's again, reasonable fit. We can look at some individual plots. We have our model and our fit. That takes a second to bring those individual plots up. One might argue that we may still have some missing covariates, right? We look at our preds here and they don't really match for a couple of patients, but this is the model that we're going to use for our simulation. So what we're going to do in the example is we're first going to take this model and run a simulation for a single dose, right? And then we're going to modify that to run a simulation for a steady state dose regimen. And if we have time, I'll show how to do a multiple dose regimen as well, but we'll see. I think I'll have time. So the first thing we're going to do is we're going to take our final data set, right? And we're going to add a column with a mutate statement called steady state, where if the amount is greater than 0, again, that's the dose records, we're going to assign a value of 1, otherwise leave it as 0. And then what I did is I cut our original data set, which had 150 subjects, down to 10 subjects, just so these examples run quickly for the demo. If we look at our sim data set, right, we see our steady state. That's the only change that we have to make to the data set. Next, we're going to set up a simulation as we covered in lesson 7. So how do we do that? We copy our fit model, accepting all the effects to get the parameter estimates into the model. And then what we're going to do is replace that original data set that was used in final model with our sim data set. If we just copy this model and then check the input data for sim mod, right? The sim mod is our simulation model. If we look at that right now, we see it's still got 150 subjects. So in the past, we've showed how to use data mapping to replace that. It's also possible just to directly replace that component of sim mod with our new SimDat data set. So if we go ahead and do that, this is a shortcut, so you don't have to go back through the mappings procedure. And if we just look at the input data now, we see there's 10 subjects in there. So now we can print that model just to check the mappings, see that everything's okay. Now note that there's no steady state mapped because we haven't told the model about that steady state column yet. Again, at this point, we're just going to simulate a reference single dose profile that we'll later compare in a graph to our steady state profile. The other thing I did here is I cut down the random effects to a very low number, just so we could see, you know, with some more delineation, the difference between steady state and single dose. Here we define our simulation table. So we're going to generate a simulation output called simtable.csv. We're going to have time output from 0 to 48 by 0.5, and these are the variables we're going to output. And we will do 10 replicates. I have a random seed number in here, and we call the simtable object here into the simsetup NLME simparameters command, which we will feed into the simulation. Again, I've already run this simulation, so I'm not going to re-execute that. This little function right here, it's worth mentioning. It's a function that simply checks for an existing simulation directory. So here I have like simmod because I've run these before. In the current version of RSNLME, which we are correcting this, if you try to overwrite a simulation folder, it will not overwrite. So if you're in a situation where you're trying to run simulations and you're making changes like replicates to 100 and you're only seeing 10, just go ahead and delete that folder and rerun the simulation. Perhaps that's already happened to you, but this is something that we are fixing. This is just simply a little function that I wrote to clean out that directory before executing a new simulation. So again, I won't run this now. I'll just go right to the plot, and we have a single dose. If I go to my plots, we have a single dose simulation for my original model, C versus time single dose. All right, so the next thing we want to do is simulate a steady state model. So again, I'll go through this pretty quickly. We copy our model to simmod steady state. We're still using our original data set or our simulation data set, so we don't have to worry about mapping a new data set. But here's the add dose cycle command. And we're going to say simmod ss add dose cycle steady state amount AA interdose interval 12. And again, what is this doing, right? If we go and we look at our simmod ss and we peek at the column definitions, right? We have this in there now. That's what's instructing the model to run steady state simulation. And you also see how the simulation table gets input into the column definitions. So yeah, this call definitions is kind of unique about PML, and it's a very, very powerful tool for kind of thinking to do something and then just implementing it. Instead of only having your data set and your model to change scenarios, you have the column defs as an additional lever to achieve what you need to do. So I think the rest of this is just overview. What I'll do is I'll just run to the actual plot. So here we have our steady state plot, and sure enough, it looks like we had some accumulation, right, compared to our single dose situation. And that's how you would use column definitions. So I hope it's a useful example of the concept that Mike described in terms of being able to think of something different and then implement it very quickly. The remainder of this code actually shows the use of the add dose cycle for an additional dose. So this would be for a multiple dose regimen. And if I just jump to the plot, what I've done is I've plotted a multiple dose regimen where I gave 10 additional doses, and then I superimposed the steady state curve in blue, just as a check to see, all right, is that steady state simulation reasonable? And well, yeah, it looks reasonable. So yeah, that's the end of my demonstration. I think we have we wanted to leave some times for questions at the end. Before we go to questions, I just wanted to again plug for lesson 13. Lesson 13, we're going to take a look at hands-on application of PML. So in that lesson, we're going to take a one-compartment model and we're going to create a anaerobatic recirculation model using the code that Mike had showed a bit earlier. So that'll be a good one. We're going to talk a bit about the sequence and sleep statements, which kind of difficult to get your head around, but give you a tremendous amount of control in terms of being able to turn things on and off in models. So yeah, PML is, again, it's a declarative language for pharmacometrics that has a tremendous amount of capability to control very complex parallel tasks, in addition to, the normal modeling stuff. So I'll stop there. Thanks for your attention. And at this point, I think I'll turn it over to Anna for questions.
Yes, well, thank you to both. Great, great presentation. And we do have a couple of questions. And if others have more questions, please feel free to put them in the chat. So the first question is probably better to address it for Mike. The question is, what is the difference between theta and fixed effect? And also the difference between omega and eta and random effect. So we see that PML uses fixed and random effects, but modelers prefer the theta, omega, and eta terms. So if we can clarify that.
Hi, the history of mixed effect modeling said that it comes from non-mem, and in non-mem the fixed effects are called thetas, and the random effects are called etas. So you can say it's a mistake or something, but I've basically confused the two, where we say finding the best thetas is finding the best fixed effects, and finding the best etas is finding the best random effects. Now, what the variance is the fixed effects are point estimates, but the random effects vary for each subject. They're different, and so what we want to do is get an idea of how the random effects are spread out. Now, if the fixed effects are correct, then the average values of the random effects should be 0. So, but they are spread out, and that's the amount by which they're spread out is called the variance. So that's why we're interested in it, because we want this to work with subjects who are not in the population that we started with. So does that answer the question?
I believe it does. Thank you. We have one other question, maybe this one for Keith. Do you continue to expand the PML language, or is it going to stay as is?
We continue to expand the applications of the language. As I mentioned earlier, we've had to make very few changes to the language itself. Again, one of the things that I'm continually, amazed by, that this was developed 20 years ago, and we're still thinking of new applications and haven't had to change the core of that language at all. But certainly, yes, we're releasing, new versions of R. Piranha probably once or twice a year. We have a release in November this year that will include an R package for running machine learning assisted model selection using NLME and PML. So yes, we continue forward.
If I can contribute a little bit on that, there were points in the past where we made improvements to PML. One of them is to add the steady state capability, and if you think about it at all, that's a pretty complicated thing to do. Another was you can have up to 5 levels of subject ID and 5 levels of random effects, and I've never seen anybody actually use that. So, but for instance, you might have center effects, which all the subjects in one center are different from all the subjects in another center. Well, that would be another level of subject identifier. So I'm trying to think. There are other. Let's see. We added various forms of delays. If you wanted to have a delayed signal, delayed by a certain amount or delayed in a decreasing fashion, that's another property that was added along the way. But I think it's, these are things that I've never seen anybody actually use them.
Great, thank you. We have one more question, probably have time for it, is how would you decide to work with our speaks and LME, with RS and LME? So for example, I think what it's trying to say is maybe you can give us an example of moving from Phoenix to using RS and LME. How do you decide that maybe that's something that you need to do at that point?
Well, I think the answer, and I hate to say this because as a developer, we hear it all the time and it makes it difficult to build, hard-coded solutions, but it depends. Generally, one would reach for R speaks NLME if they reach a point where something cannot be achieved within the Phoenix GUI. And there you have a choice even, right? You could use the R shell script object within Phoenix to implement some custom capability. But if you get proficient with R-based solutions, there's a tremendous amount of efficiency that can be gained. You could have a single R script that contains your whole model development process. And as we've shown in some of these R school lectures, you could be building a markdown-based report in parallel to that. So I think it's in a way, it's an individual decision. There is the cost of, R being a more complex environment to work in, but the gain is the level of efficiency that you can achieve, and also reproducibility. And again, the reproducibility stuff, probably the best examples are the simple ones, right? You may have your population report if you generated a Word document, right? You copied a bunch of stuff from your Phoenix outputs or maybe some custom R plots, you send it out to your team for review, and the marketing group comes back and says, you know what, we don't want to use the drug number anymore. We want to use the trade name. And you look at your 150 plots in your report, and well, now you have to go back and change that, one at a time. If you built that report in R, really it's a search and replace in the code, and then you regenerate your report. You could be done in probably an hour compared to perhaps days. So again, maybe a very simple example, but it's these kind of painful activities that kind of drive people in some respects to the use of these command line tools, these scripting languages. I hope that helps answer your question. Yeah, but it's easy to go from Phoenix to RSNLME. There's the textual model function where you can just import a PML model and a data set from Phoenix and immediately run it in R with no changes to the model.
Great. Well, thank you very much. This will bring us to the end of this webinar. We thank you, our speakers, for being with us today and for everybody who attended. And we hope that we see you in the next lesson. Thank you. Thank you very much.
Thank you. Bye-bye.